And we are now being recorded. And the bad news about single neurons and the bad news about the nervous system in general is that we know very little. And so I'm going to start by going into the context of both what we know, but more the context of what people think we know. And I have to say that, unfortunately, I think we as somewhat bottom-up people, as people who pay attention to the details of biology, remain in the minority in computational neuroscience. I also, as I said earlier, crave questions. Questions make me feel like people care, people paying attention. That makes me feel good to just ask a question from time to time. Now, unfortunately, I cannot see the raised hands, right? Because I'm seeing my screen, or I can move to here, and then I'm seeing, but then you're seeing this. What are you seeing? See, now I crave a comment. What, do you see my presentation now, or you see yourself?
We see you and all the other participants, but we don't see.
So that's not helpful. So I cannot see both. So I have to look at my screen to see. Oh, it's just way too much for me. As you can see, I'm old, and you know, all this stuff is new to me. I used Fortran, assembly language, machine language, and now it's Python and Zoom and Google Meet. Okay, so this should be my second slide. Indeed, it is. And our approach, by our, I mean people like Terry Sanyovsky, who I trained with, although he actually takes a dual approach. He's very much into artificial neural networks. People like Gordon Shepard, who Robert trained with in part, Michael Hines, who both Robert and I trained with in part, and the whole group at Blue Brain Project, Human Brain Project, and a lot of other people who you'll hear from, in fact, as little presentations next week, and whom I don't have time or I can't think of to name, but maybe I'll try to bring up some names as we go through, believe that computational neuroscience is part of neuroscience. So that if you do not understand the details of neuroscience, you will not be able to build models that are realistic, that are helpful, that are useful for understanding the brain. So this week, obviously, you're going to be learning NetPyNE, and that is going to be your full-time job. So this is not the week to look at these textbooks. But if you are interested, and if you're not already a neuroscientist, and some of you, of course, are, then these are some of the books that might help you. And I just picked out the ones that I'd heard of, frankly. There may be better ones out there. The book that I used as a student was Kandel, Principles of Neuroscience, and actually it was a student at Kandel's institution at Columbia back before the textbook was written, so we worked from the notes that became the textbook, but it was really the same book, although back in the 0th edition, version 0.1. Purvis seems very good, and I noticed looking at some of Salvador's slides that he helped me set up this talk by giving me his slides, that they have a wonderful website, Neuroscience 5e at .Cinauer, where they have these animations that are great for looking at things, so that's recommended. Bear is another popular one. HILA is one for understanding the lower level details, so ion channels. And the third or fourth, whatever edition it is now, has become very fat, and maybe not be the place to start, or you'll just get intimidated and freak out. The place to start, in my opinion, is the first edition, which you can get, luckily, online, and just look through for his explanation, for example, of Hodgkin-Huxley, which we're going to hear a lot about Hodgkin-Huxley. Those are the guys who came up with the idea of the mechanism underlying the action potential. And so they're very important in this kind of bottom-up, basic detail, computational neuroscience. Nichols is also a popular book. Again, there are many other books. The nice thing about these books is that they will tend to agree on the concepts of neuroscience, at least up till you get to cortex, consciousness, decision-making, some of the higher level things. Now, the agreement is both a good thing and a bad thing. I mean, it means that people are not, to my mind, exploring enough. But of course, this is, textbooks for lower level graduate students and for undergraduates. And so you have to give a story. And so to some extent, I will try to give a story today. The bad news, as I alluded to just a moment ago, is that when you get to computational neuroscience, you have no agreement. And so right now, and this was a set of books I got pictures of, little icons of, a couple of years ago, 3 or 4 years ago, I think now, when I one of the last courses in Brazil. The most popular right now is deep learning and convolutional neural networks. And you'll see from the phrase neural networks that the people who work on these, that design these, Jeff Hinton and Terry Siniavsky and others, certainly many others since then, Jan LeCun, believe that this has something to do with neurons. It does not. But it's not unreasonable to think about and consider the kinds of concepts that one finds out from deep learning or from Hopfield networks or from other really abstract models, but it is a mistake to take them literally. And I'm going to harp on that notion quite a bit, I'm afraid. This is the Neuron book. Neuron is the simulator that NetPyNE uses. This book, unfortunately, is a bit obsolete. We put in a proposal for a second edition. We got knocked back. That was distressing because, again, we believe that this type of approach is the way to understand the brain, to understand neurons. And the people in ArcSpec said, well, we already understand the brain because we have read Peter Diane's book, which is here. Are you seeing my pointer, by the way? Or do I have a way to make it bigger so that it's more visible? Because I can barely see my pointer. Let's see, is there something that makes a pointer bigger? What did that do?
Yes, Bill, we can see your pointer.
Okay, good. So this, I think, is the best seller of probably of all the books here, maybe. And Diane and Abbott, widely used. And I'm embarrassed to say I cannot get past their introduction because the assumptions it makes are, to my mind, not warranted. And the particular assumption, the assumption of rate coding, the assumption that you're going to replace the neuron with a scalar approximation of neuron state. And as was somewhat alluded to, what's the neuron state? Calcium level, is that the neuron state? Calcium level in spines, perhaps that's the synapse state, and so perhaps there are 10,000 neuron states that we need to deal with. Is just sticking with voltage, what's the neuron state? Is it the voltage or is it the spiking? Rate coding would suggest it's just the rate of spiking, and I think that's a mistaken assumption. Some of these other books make similar assumptions. This is more a mathy book by Izakevich, which is very interesting, very cleverly written, but again, very abstract. This book, I don't know if this has reached another edition, Methods of Neural Modeling. It tends to be more detailed, which I like. Again, all I'm giving you is my personal prejudices. Here's a book which I don't think I've read. Some of these, this is a very high level, Eliason's book about modeling cognitive processes using very, again, simple scalar type things. This is a classic, Perceptrons by Minsky and Papert, which led to the notion that backprop and what now is deep learning convolutional networks could not work. Turns out they were wrong. David Marr remains very influential, the 3 levels. And I'm sure a lot of what I'm saying here is not understandable because you don't know what 3 levels are. If anybody, if I touch that, it goes to the next screen. But if anybody's very curious, they can ask, or we can discuss later on Slack. Here's my book, which is also somewhat obsolete. It's now 20 years old. But part 4 is the part about brains. Parts 1, 2, maybe 3, I guess, are about computers more and really not relevant to this course. Okay, so it's a Tower of Babel. It's so many different languages, people speaking to try to understand the same thing, the brain. And we're in the situation of the proverbial 6 blind people and the elephant. One person feels the tail and says an elephant is like a string, that feels the trunk, says an elephant is like a snake, feels the ear, says the elephant is like a leaf, feels the leg says the elephant's like a trunk. There must be two more, but I don't know what they are, but that's the idea. So I divide up these paradigms, or really, you could also say fads, because people just glom on to the thing that suits their training, frankly, to decide how are we to best to study the brain. So we have these three strands. Computer science strand, probably everyone else familiar with because of the fantastic practical applications of convolutional deep learning networks. And that came out of the backprop revolution, such as it was in the 1980s, and parallel distributed processing in the 1980s, and has really come into fruition. The physics math approach, I'd say the Hopfield network would be the most prominent exemplar of that approach. That is a type of network that does a memory system based on auto association across vectors, basically dot products across vectors. And the physics and math folk tend to work with theories. And in fact, we have an ongoing collaboration with one of these physics math folk, and he insists, well, we're going to assume the dendrite is a symmetrical object around a spine. And I don't think it is. Well, of course it's not. And I don't know how much you can learn from assuming it is, but we argue about this. But, you know, if you're given lemons, make lemonade. And we consider ourselves, perhaps a bit arrogantly, to be biology. We work with experimentalists. Some people in the field actually do experiments. I do not. And we try to make, sometimes with less success than other times, direct predictions of what the experimentalist will expect to find. And on rare occasions, they have actually found it. And we've been very pleased and talk a lot about these successes a lot. So if you want to go to meetings, the meeting we'll send you to is the one at the bottom. The biology type meeting is cns.org. CNSorg.org is their handle, and it takes place in July. I think if you want to submit an abstract, the abstract submission deadline is the 21st of May. Just got delayed, so it's soon. And so that's the one which has a fair amount of biology, although, to be honest, a fair amount of stuff that's higher level, more hypothetical, more theoretical. Cosine, going from bottom to top here, is also quite biological, but self-identifies as being a meeting about applying the latest ideas in engineering or statistics or control theory, other fields of engineering and physics to the nervous system. So they work with experimentalists, but rather than trying to build from the bottom up at all, they work with techniques that they can then apply to data sets. And in doing so, they, like us, like all of us, make a certain set of assumptions. And to my mind, the assumption that I'm concerned about is they assume rate coding in many cases. And that's, again, an assumption I think is not warranted. Going to the top one, what used to be called the NIPS meeting, and it's still nips.cc, but apparently if you search for NIPS, which I didn't dare do, you come up with discouraging returns, and so they changed the name to NeurIPS. And they're the most computer science. They're the ones, Terry Sanyovsky was in charge of it early on and was trying to include biology, but that has not really happened. It really became the backprop meeting. And now the deep learning meeting, Jeff Hinton, Jan LeCun, that'd be the Facebook and Google guys, go to that. And that meeting is in December. And I think that meeting is, yeah, you've already missed the abstract deadline for that. It's pretty hard to get an abstract in, frankly. Cosine meeting, the abstract deadline is not for a while. So 3 kinds of simulators, 3 kinds of ways of looking at maybe the brain. So computers, now I'll start from the top, go to the bottom. Computer science folk are TensorFlow would be, I think, the main program for dealing with deep learning networks. That comes, I believe, out of Google. Actually, when I started, again, many years ago, the program we used to simulate neurons was SPICE, which was an electronics program, which I think is still active, but more for developing analog electronics, things like antennas, transformers, things that you would see. Sorry, I got interrupted by. Do you guys see that little thing? It doesn't matter, actually. Analog electronics. Then Magic, I think, is the open source example of one of the VLSI tools. So for developing very large integrated circuits. And these would be things that you would use in electronics and CS. Physics and math, people tend to use MATLAB. If you're using continuous equations, you might use Simulink. Mathematica also is a good tool for that. In the area of finite element analysis, discrete voxels that interact with other voxels next to them through fluxes. You might use Phoenix, which is the free one, or COMSOL, which is the expensive one for that. So again, we are biology, so we use NetPyNE, which we're teaching here, and Neuron, which we'll also teach to some extent, because if you want to modify NetPyNE to do something that hasn't been done before, you go down to Neuron code. And hopefully you don't have to modify Neuron, because for that you have to go down to C code, but if you, or C++ code, as well as Python code. But if you do, we certainly would welcome you to join the Neuron developer group, which is active in that. It's NetPyNE developer group and Neuron developer. Genesis was very popular for years, is not being actively developed at this time, so I'd say is become less popular, but still usable. Moose is a project out of India by Upi Bala. It has gotten off to a slow start, but it has the idea also of doing multi-scale modeling comparable to what we do with NetPyNE. If you want to go into multi-scale musculoskeletal work, there's OpenSim. I'm just really giving it a few that I know about, because there are no doubt many more that do this, and there are no doubt many that do liver or do immune system that would actually be potentially interesting if one is doing diseases of the brain. Interacting with the immune system, obviously very important. Interacting with vascular system, very important. And these all have their own simulators that we talk about linking up, but in fact are very hard to link up to get a more full picture of the brain. To go down to a lower level, the level that will present as reaction diffusion, the RxD module in Neuron, we have some simulators that really work primarily at that level, or in the case of MCell, really just at that level. MCell actually simulates individual atoms down to the level of an individual calcium ion traveling through tissue. Its flight through tissue, well, through the extracellular space, and banging into membranes and into things that it can combine with, such as calmodulins. It can do extracellular, it can do intracellular. Steps would be another one, a little bit broader, going up to the level of a cell. And then more general cell reaction diffusion would be VCell, comes out of University of Connecticut, Les Rose group. And libRoadrunner is just one of many interpreters for SBML, which is the Systems Biology Markup Language, and that comes out of University of Washington, Herbert Soros group. So, key point, again, is that your prejudices, your fad, your priors, your intellectual prior determines how you're going to think about a neuron, about the brain, about a synapse, about a dendrite. And so you are a prisoner of your thoughts, as you know, as we all are. We are prisoners of our perceptions. And ideally, we want to make those explicit, but sometimes they are implicit and we don't even know what we're thinking And to some extent, that's actually an advantage of simulation technology. You don't know what you're thinking until you get it into the simulator and you see, oh, well, those are odd consequences, and those consequences come from this assumption that I never really thought about fully. So a simulation can be useful for that. It's the same idea as people writing a book. They say, I write in order to know what I think. Once you get it down on paper, it's suddenly much more clear what your assumptions, how your thinking is working. So we do a computer model to instantiate a conceptual model. So we have to have a concept of what the neuron does. And again, I'm going to come back to this again and again, that our conceptual model of the neuron dates back to the 1880s and 1890s, which does not mean it's wrong. I mean, that may be a very good conceptual model, but it's, to my mind, clearly a partial conceptual model, and it's remarkably archaic. I guess there are other sciences that harken back to the 1890s, and Maxwell must have been back then, right? And he developed field theory for understanding electromagnetism, and that's still very useful and used. So, but just from the technology, you can readily tell that what Maxwell said is correct and very useful. Whereas in neuroscience, as Obama said, as Salvador quoted Obama as saying, we still don't know shit. Did Obama say that? Not exactly, but that's kind of what he was implying. And he, of course, had developed that notion by talking to a group of people that, what just got unplugged is my power. Talking to a group of people who are neuroscientists who said, you know, we don't have the basic measuring devices to understand the brain yet. And so that's what the so-called Obama Brain Project, BRAIN is an acronym standing for Brain Research Advancing Through Innovative Neurotechnologies, is meant to do, is develop the innovative neurotechnologies. And in fact, we present, when we do a grant with the National Institute of Health, we say NetPyNE and Neuron are themselves innovative neurotechnologies for understanding the brain. So, can we get new conceptual models? Yes, we can. And you can get them from new experiments where you have, you know, eureka, aha, voila. We have a moment where we suddenly now understand something. And those moments can also come from new modeling results, that something doesn't make sense. And generally, when it doesn't make sense, you track it back and you discover, hey, there's a bug in my code. And then you're very disappointed because your Nature paper turned out to be a bug in your code and you can't publish it. But sometimes, and I'm not sure I've ever actually experienced this, but I hope to, you'll come up with a new idea from your modeling. I mean, most of the time, your modeling confirms some of the assumptions you had, and I'd say, I'm being a little unfair to myself, expands it. So you do get some new notions. There is some added value. But in terms of, wow, we've got to replace Cajal's 1880, 1890 theory with something new, that hasn't happened to me yet.
Bill?
So we're going to talk about the standard model of the neuron, Cajal's neuron doctrine.
Bill, we keep seeing
Yes.
your Slack messages, even private channels and everything, so we don't want to see anything embarrassing.
Embarrassing. Okay, yeah, that's going to get worse and worse. Let me see. So I can just kill Slack, right? Okay. Did anything embarrassing already happen? Don't tell me. I'll just be too embarrassed. Okay, so there comes the train, which is noisy. Can you hear the train? No? Okay, yes. Good. Let me wait for the train to go by. So the standard model of neuron, Cajal model, Cajal neuron doctrine, signals come from connections. I don't think he called them, I'm sure he didn't call them synapses. Of course, he spoke Spanish, so I don't know what he called them. And connections between neurons, that was part of Cajal's viewpoint and part of his brilliance, that he realized that neurons are in fact distinct cellular entities, as against Golgi, who thought that they were all a syncytium, like some of the cells in the heart are. Input comes to dendrites, Dendrites communicate with soma, soma communicates with axon, axon communicates with the next cell. And this is a standard doctrine, a central dogma almost. Eccles was the one who got the Nobel Prize. So Cajal got a Nobel Prize. Eccles, decades later, got a Nobel Prize for saying that synapses are mostly chemical transmission and not electrical transmission. And he's right and he's wrong because they're both. And that's the other thing about biology. Whatever rule you get, whatever dogma you get, there are going to be exceptions. In fact, the central dogma, as people know probably, of DNA was DNA makes RNA makes protein. That's what Francis Crick sold as the central dogma. And it worked for many years until we discovered that mRNA can make DNA. And in fact, Crick adamantly rejected that, and got in the people who wanted to publish it had trouble publishing it, and they got into a bit of a spat with Crick, Francis Crick, and they eventually got their Nobel Prize. So there. But dogmas are made to be broken in biology. In physics, you can say, okay, Maxwell's equations are what they are, and maybe, well, anyway, I don't know enough about whether they hold for dark matter and dark energy or not. Probably they don't, but who knows? Anyway, so this rate coding notion of scalar encoding is, to my mind, very limited. It's not wrong. Biothinking can be true. Rate coding can be true. You can get information out of the rate, so there's something encoded in the rate, but it still can be wrong. And that's, central dogma molecular biology. Could be wrong and true, which is funny. And if you're a physicist and you're an engineer, that's uncomfortable. Things that are true should be always true. Salvador mentioned multi-scale modeling. This is just a different figure saying the same thing. I think the thing that makes our organ, the brain, and even the spinal cord, not so much perhaps the peripheral nervous system. Whoops, if I touch that, it moves. I gotta be careful. Different from the liver, the kidney, maybe the immune system, so that's debatable, is that there's so much overlap between scales. So here we have time on the x-axis from microseconds up to, in effect, we would want to take this up to years because development happens over years and decades, and spatial down from nanometers, although actually it seems we just, that's not, that's okay, I guess it's got microns anyway. Microns we have here, but it should be down to nanometers up to the size of the brain, which is maybe order a meter, well, half a meter, quarter of a meter, call it order a meter. Many levels of modeling, many scales, and particularly in the area that NetPyNE is interested in and Neuron is interested in, we have this enormous overlap between the cell layer and the cell scale and the layer of cortex scale, or going to different parts of the brain, the regions of thalamus, the pieces of the basal ganglia, anterior and dorsal horn of the spinal cord with their layering. All of these are so complex, and there's so much interaction between scales that we cannot simply encapsulate the cell, as again some of the paradigms would do, and say, this is what a cell does, now let's see what a network does using those cells. I tend to think you can do that with the liver, and you cannot do that with the brain. But I have not done liver modeling, so I may be being unfair and oversimplifying the liver. Neurons, as well as liver cells, as well as immune cells, they're all hacked cells. They all make use of what was originally available to whatever the core life form was underwater, which then became some kind of archaea or prokaryote. There was actually a recent paper as to whether that was likely to have been double membraned or single membraned, and then grew up to become eukaryotes. Maybe we could think about yeast as single cell eukaryotes, various protists, and then multicellular eukaryotes such as ourselves with extreme specialization of cells. So the neuron is a highly specialized cell, but it still has to work. First of all, it has to do all of these regular sorts of things that any cell has to do. It has to metabolize. It has to grow. It has to be able to divide, at least at some stages. And there's actually even evidence that neurogenesis occurs in the adult. And so it utilizes aspects of what it's doing anyway in its processes for signaling. So something like a membrane potential, which we're going to talk a lot about, is akin to what a mitochondrion does to create ATP. It creates it out of a hydrogen potential difference across the membrane. The calcium activation, a lot of it's due to calcium-induced calcium release from endoplasmic reticulum and to some extent perhaps from mitochondria. And so endoplasmic reticulum, which is an organelle which has a lot of important usage in terms of creating proteins, rough ER, smooth ER, is also involved in signaling. When you think about the types of molecules that are used as neurotransmitters, you think about things like GABA, which is gamma aminobutyric acid, which is some kind of spoiled butter. So you are what you eat. You think what you eat. Glutamate, which is umami, which tastes good. Presumably GABA does not taste good. Oh, I haven't tasted it. So all of these complex things have to emerge from cells. They're not designed from the top down like an engineer would do. There is no God. Sorry. No, I don't want to get into that. I don't want to make any enemies. There may be a God, but it's pretty clear that he or she did not design the neuron because it's really created from bottom up. It has all these pieces that are present in any cell, and it reutilizes them in very clever ways that we don't understand and that we want to understand. So what does a neuron do? What's a neuron for? What is it? And this is a mystery. And why do I want to present you with mysteries instead of with knowledge? Because we have far more mysteries than we have knowledge. We have far more that we do not know. And again, I come back to Obama and his committee that said, we just don't know. We don't know what a neuron does. We don't know how it does it. There are enigmas and mysteries, and enigmas wrapped in mysteries are wrapped in puzzles. That's a paraphrase from some famous quote. Why is this happening? I decided to use the little tag word "huh," as I should have used pound "huh," hashtag "huh," to signal when I've got something I don't understand, and I'm pretty sure nobody understands. So we think of neurons as being grossly a feed-forward element. Of course, people who do deep learning, who do backprop, also think about the backward aspect of that and would like to assign that to, for example, the cannabinoids. But there's really no evidence that cannabinoids can do anything like what backprop does. What are the dynamics of the neuron? Well, when you talk about dynamics, most of what we're going to talk about is going to be electrophysiology. It's going to be the interaction of EPSPs, excitatory postsynaptic potentials, IPSPs, inhibitory postsynaptic potentials, action potentials, other potentials. But that's the tip of the iceberg because you have all this calcium, a lot of even protein synthesis going on that's interacting, again, scales on top of scales, wheels within wheels, to create dynamics at different scales that cannot be segregated, cannot be separated from each other. Are action potentials really all or none? Well, I would say yes. For practical purposes, for what we want to think about, let's say they all are all or none. But you have a famous experiment by David McCormick where he slightly depolarized the cell body and showed that now when the action potential, and I hope I'm getting this correct, when the action potential arrives at the presynaptic terminal, which is miles away, figuratively speaking, you have more release of transmitter. So it's just a little more going on there than just the all or none signal transmission. Another example, I had another example in mind, and now it's not coming to me. It'll come to me in a minute. I'll come back to it. I already spoke of the importance of chemophysiology. It's generally been ignored in neurons. Obviously, people who do liver cell thinking and modeling, they think a lot about chemophysiology, but we tend to not. Calcium is the one thing. Avrama Blackwell says calcium is the answer to everything. Perhaps it is, but that has been explored some. Cyclic AMP, G proteins, these have been explored, but not really very much in modeling, and they're clearly very important. Degeneracy. This is a notion, I think the original paper would have been Edelman and Gall, forget the name, and now is very, very much taken up by Eve Marder and her students. The idea being that not only do our models not converge on a single set of parameters for explaining the dynamics that are observed in a cell or network, but that even the organism, and Eve Marder studies the stomatogastric ganglion of the crab, even the organism and that little piece of the organism cannot come up with a single set of parameters and does not come up with a single set of parameters. There is a degeneracy. There are multiple solutions to the problem of generating the dynamics that are observed. So you don't have a single answer to work with in the way that you typically do in physics and engineering. You have to say we have a population of cells that are identified as being the same cell, and in some of the invertebrates, in particular in stomatogastric ganglion, you can identify a cell very precisely by its position and by its dynamics. So you can say this is a P cell, and having said that, you can now say, okay, well, what's the solution for how a P cell behaves? Well, there's not a single solution. And Eve Marder has pointed out this is actually a feature, not a bug, because it gives you more opportunities to survive in evolutionary terms in the context of alterations in environmental circumstances. The fact that you do not put all your eggs in one basket, that you have many different solutions, many different ways of doing things, means that you can identify, you cannot identify a particular solution. You can survive. Some of these organisms can survive even when things change drastically. In the case of the crab, saline changes, temperature changes, other changes. The synapse is a very complex thing. Endocannabinoids going backwards, volume conduction by gases, NO particularly, co-transmission of peptides and GABA. What's a modulator and what's a neurotransmitter? Modulators are more widespread. Gases are most widespread. How widespread? We don't know. Synaptic transmission, extrasynaptic transmission. Synaptic nucleus communication. One of the ideas there is synaptic tagging and capture, that if a synapse needs to learn something, it has to identify itself to the nucleus so the nucleus can send up the proper mRNA to the proper synapse to get augmented or decremented. So that would just be one example, but there's evidence that NMDA transmission, which is a classic fast transmitter and through its ionotropic effects, is also something that allows a synapse to communicate with the nucleus through some of its metabotropic effects. Ionotropic means letting in ions, ions, and metabotropic means changing, in some respect, the metabolism of the cell, such as different expression through from the nucleus of mRNA, or often more locally, just changing these protein cascades, which are viewed as they are not actually metabolic, but they're viewed in the same way. They're chemophysiology. Spines are very interesting and mysterious. Is a spine an electrical apparatus or a chemical apparatus, or more likely both? Why do they look so funny? They have these different shapes. Some of them look like mushrooms, some of them like lollipops, some of them little stubs. They grow and they change with learning. So mystery, 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 mystery. So the first mystery for you all to understand is the mystery of how to write programs in NetPyNE. A second one would be how to write programs in Neuron. And as I say, that's going to be your occupation for the next 2 weeks. But I will present you with at least some of the basic concepts of neuroscience, and this would be one of the most basic, and I just written it out in as kind of compact a way as I could. These are the famous Hodgkin-Huxley equations. So this is capacitance, and this is the first derivative of voltage with respect to time. This is the maximal conductance for a sodium channel. Na is sodium. The over bar here means maximal. It's called G bar. Oops. G is conductance. These are what are called the particles of the sodium channel, the M particle, which really doesn't mean much, the M gate to the third power, the H gates to the first power. This is the voltage of the membrane, so that's the same voltage as here, but this one's, of course, first derivative. This is zeroth derivative, non-derivative. This is the reversal potential for sodium. Similarly, here for potassium, this is the N gating or N particle. Similarly here for the leak current, which has no particle. Each of these, M, H, and N, are described by this kind of equation. I just wrote X because they're all the same. So M dot would equal M infinity minus X over tau M. H dot would equal H infinity minus H over tau H. And then these are defined in terms of alphas and betas. Now, why would I let's start with this. First, what are you to think about when you see this? If this is your first time seeing this, of course, this is total gibberish. That's understandable. That's to be expected. Do not panic. Do not freak out. You have to see this. I had to see this, I would say, 7 or 8 times before I felt like this is obvious. The third time you see it, hey, I've seen that before. Not even the second time. I don't remember having seen it before. The third or fourth time, it's familiar, but what the hell? I still don't know what the hell it is. The 7th or 8th time, especially if you've gone through a bunch of exercises, done the graphics, shown why alpha and beta relates to infinity and tau in this particular way. All these are wonderful exercises beyond the scope of this 2-week period, but something we could think about doing later. Eventually, 7th or 8th try, 10th try, this stuff becomes obvious. Even I do not remember that the exponent here starts with negative 0.0556. Even I, who've been doing this for decades. So there you go. Obvious does not mean I've memorized it. I could memorize it, I suppose. Anyway, why worry about it? Who cares? The computer does it all for you. You don't have to do anything. You don't have to think about anything. It's all handled for you. It's like in elementary school, why learn addition? The calculators do it for you. Well, you could argue that because, yeah, addition, well, I don't know. Anyway, I think you should learn addition. You probably have. So the reason to worry is not because you need to type this in, but because these models are all limited. Your simulation is not your conceptual model. In particular, the conceptual model, since the work of Sockman and Nair, who showed that the ion channels postulated by Hodgkin and Huxley actually exist, they also showed something that Hodgkin and Huxley never would have guessed, that these follow a random pattern of opening, and that these can be described by Markov models. So this Hodgkin-Huxley model, like every model in biology, is wrong, but useful. Depending on where you want to model it, whether you want to model the axon, whether you want to model a point neuron, which is another questionable concept that we must use because we can't always do anything more complicated. It's important to understand the limitations of your model. It's like a famous movie quote. Clint Eastwood in Dirty Harry. A man must know his limitations. A model must know its limitations. So that is why you need to know what lies behind the modeling. where it's going to make sense and where it's not going to make sense. Oh, I wanted to give another example of this. People tend to do basic science, especially if they're only at master's level, but unfortunately even at PhD level, learning how to use a statistics package like SPPS, like a statistics package, and then they plug their numbers in and they say, okay, we have a significant result, p less than 0.05, because the program told me that. Well, okay, but if you happen to be one of these people who's hard at work trying to find a nice result, and you happen to have done 20 different experiments, and you finally find one that shows you that there's a 1 in 20 chance of having a valid, an invalid result, the null hypothesis, then you're not being fair. And that's because you have not understood what you're measuring. You've not understood how it works. And so it's important to understand what lies behind the software. So this is just showing these great men. I should move that slide up since I already talked about them. They received the Nobel Prize, 1906. Their work, in particular, Ramon y Cajal's work, is still what we use, what we think about, how we think about cells, which again is remarkable because it's a long time ago, and we ought to know a lot more, I'd say. Golgi was the guy they kind of fought. Golgi invented the technique. Cajal used the technique and then got the results that turned out to be correct of these cells being separate cells. But Cajal, despite brilliantly coming up with the notion of a neuronal dogma, dendrites give messages to somata, give to a soma, give messages to the to the axon. Sorry. He also showed that neurons are so variable, and we now assume, but still do not understand, how they work in very different ways. Purkinje cell, misspelled. I wonder what book this came out of. Purkinje cell is cool. One of the things that's interesting about the cells of the cerebellum, in particular Purkinje cell, which is principal cell of the cerebellum, and the spiny stellate cell, is that represented here somewhere? I think not. There's a globus pallidus cell of the basal ganglia, is that these are inhibitory cells. So we tend to think about networks of neurons, in particular when you work with deep learning and cortical models, you talk about networks of excitatory neurons. And yet these are major brain structures where the principal cells are inhibitory cells. So that's odd. And what, how, why? I don't know. I wish I did. And that's the other thing. You guys are young. You will figure this out. And all I ask is, during your Nobel Prize acceptance speech, please mention me. Long dead, I fear. Neural doctor, Ramon y Cajal. Axon, synapses on a dendrite. Of course, there are many kinds of dendrites. In fact, we've written a couple papers about the importance of the basilar dendrites. We've written less about the importance of the apical dendrites, mostly because those are even more difficult to experimentally explore than the basilar dendrites. Both are difficult. Synapses, chemical synapses, dendrites, talk to soma, talk to axon. The notion of dynamic polarization. So the dynamics of the cell follow the structure of the cell in this sense. What to say? Many things can be said. There's backpropagating, not in the sense of deep learning networks and backprop, but backpropagating action potentials that start at the soma and go up to the dendrites. And those are, we assume, are probably very important, but are not well understood in terms of their implications. And there are also other kinds of synapses. So here is the importance of spikes. Spikes are important, but are most of the calculations that are being done by the brain, to the extent the brains do calculations, in the network through spiking, through the coding of spiking, or are most or many or some of them in the tree, dendritic tree, different types of dendritic trees? I would assume the latter, mostly because maintaining these dendritic trees is extremely metabolically expensive. And you would not survive as a creature spending all of this currency, energetic currency, all of this glucose, all of this oxygen on these structures, which could be better spent on muscles to smite other creatures, when you could do just as good a job with a short neuron. I think. Many people think. So I think these neurons are doing fantastically complicated, interesting things. One person who would agree with me would be Bart Mell, although I think he takes somewhat too simplistic a view in reducing the dendritic tree to an artificial neural network. So that's one way of thinking about it. Let's just check what time it is. I can see that what time do I finish? Not for a while, right? When did I start?
Yeah.
11:30? I'm already an hour.
Yeah, 25 minutes left.
Okay. And I haven't even gotten started. All I've been doing is introduction. Well, luckily I have another talk tomorrow, right?
Actually, sorry, Bill. In theory, it's just 15 minutes. 1-5.
1-5. Okay. Well, as far as we get, and then we go. Okay. I won't speed up, Salvador, because I have too much to say. I'm full of ideas. Neurons are very long. Axons are very long. They go on for miles, figuratively speaking. They go on for over a meter, literally speaking. Myelinated neurons are very common in mammals. They are not found in the squid or octopus, and they're not found in invertebrates. And they feature saltatory, which means jumping, conduction. That is to say, signals jump between what are called these nodes of Ranvier from the axon initial segment where you generate a spike over to the next node of Ranvier where you have a concentration of sodium and potassium channels such that a concentration that is able to generate a spike that then can jump across this installation, the myelinated area, to the next node of Ranvier. And so on, jumping down the axon, giving you a lot more speed and giving the fish an advantage over the squid. The interneuronal communication, we already said, is due to neurotransmitters largely, although there are also electrical synapses. Roger Traub, who's a very prominent modeler, is a big believer in the importance of axonal electrical connectivity through gap junctions as playing a role in the functioning of the brain. Most people do not accept that result. Well, accept the result because he showed it in an electron microscopy, but they don't accept that it's very important in the functioning of the brain. I remain agnostic. So one cell activates another neurotransmitter, causes the other cell to create spiking. It goes down its axon. But here's another, what about dendrodendritic synapses? So this is my favorite. I'm not going to go into detail because I've been talking too long already. But here in the thalamus, you have this enormously complex, what's called the synaptic triad, excitatory synapse coupled with inhibitory synapse, all coming into what's called the relay cell, which is the cell that's now going to transmit information up to the cortex. So you have these inputs from parabrachial region. You have the primary input. We think, well, of course, primary input because that's containing information out you see from the retina. And you have this interneuronal input. And so this interneuron may just serve, or this piece of the interneuron may just serve for this feed-forward inhibitory activation. I'm actually pointing with my finger. That's pretty silly because you can't see my finger. This feed-forward activation and the signal, excitatory signal that comes into the cell may never get to the cell body of the cell. A mystery. Another mystery. Axoaxonic synapses, chandelier cells, popular because they are very pretty. The only modeling result I'm aware of came from Rodney Douglas, and he tried to, the original idea was that these inhibitory inputs would cut off the axon initial segment, preventing the excitatory cell from spiking. He attempted to show in his model that that was not true. I think it's still up in the air. We actually submitted a proposal to do this study with an experimentalist that got turned down. But anyway, we move on. So we now get to the conceptual model of the membrane. And give me a 5-minute warning, Sandra, and I'll just wrap up anyway. The equivalent circuit is our conceptual model. Well, we have two conceptual models here. The equivalent circuit is our practical conceptual model because that's the one we can simulate. The one on the left is a more mental conceptual model, perhaps a back of the envelope, something you would draw for somebody, but by and large, we cannot simulate. We're starting to simulate a little of this in this reaction diffusion type of model, but we still would never get anywhere as close to simulating, for example, what these pores do. Pores are the holes in the membrane that allow ions through, also called ion channels. They allow the membrane to have a permeability to ions that the membrane otherwise does not have. To understand this in any detail, it requires molecular dynamics, which is way out of our reach, and really can only be done for order nanoseconds, maybe microseconds, and can't be done in the type of multiscale modeling that we're interested in, which is at least many milliseconds, and we like to sometimes do minutes and further. The membrane does not allow ions through because it is fat. And so ions which are charged cannot travel through fat, which is uncharged. They can travel through these holes because these holes it's an aqueous pore to some extent, and they let ions through. We then conceptualize this lipid membrane as a capacitor. Which, for those who have studied some electrical engineering, will be familiar. Everybody will have heard of a capacitor, but the idea is that it has these two plates, and the voltage across the plates is proportional to the charge on the charge difference on the two plates. And the pore itself is a resistor, and many of these pores, resistors, I'll just move on to the second one because it keeps wanting to do that, are variable resistors. And so this whole circuit connects the outside of the cell, which we consider called extracellular, and which remotely connects to ground, to the cytoplasm, which is the inside of the cell. And so we can now, having this conceptual model, electrical model of a fat cell made of lipid, of fat, and some protein. We can now take this into a set of equations, and we're going to see that we can then take that set of equations into a numerical approximation that will allow us to put it on the computer. So you can see, again, the distance from our conceptual model that we're going to have to go through from this picture, which is already not very realistic. Of course, it's entirely idealized. We have no idea, well, we have some idea, but very little idea what these things look like at the sub-electron microscopy level. And then to this equivalent circuit, which we have a much better handle on, but we've already made this giant leap of concept. Then to these equations, which wouldn't even be true for the equivalent circuit, although they're very good. So that's not fair. They're very good. Ohm's law is very good. People know Ohm's law. Voltage equals current times resistance. By definition, conductance is the inverse of resistance. If something has a lot of resistance, it resists current flow. If something has a lot of resistance, it has very little conductance, the inverse of resistance. G equals 1 over R. And a lot of the equations keep switching back from G to R. So I'm afraid you have to just remember G is just the reciprocal of R. So the other way of writing Ohm's law is that the current through the resistor is conductance times voltage. The law of the capacitor I remarked on earlier is that charge Q in coulombs is equal to capacitance times voltage. And so we can now find out by calculus the Q dt, you don't even need calculus, because in our numerical approximations, we just do delta Q over delta T, change in charge over change in time, is equal to capacitance, which is just a parameter, times change in voltage over change in time. And so from this, we can get a current, because change in charge over time is current. Current is flow of charge. And this is equivalent to a flow, just as x for distance, dx dt is a velocity. It's the flow of the thing along space. So current is equal to C times first derivative of voltage with respect to time. And then we can go to Kirchhoff's law, which tells us that with the conservation law, the current is conserved. So any current that you inject, we would inject it here into the cytoplasm by sticking an electrode through the membrane, or just grabbing the membrane and sucking it out with an electrode to access the cytoplasm. We could inject current here. Any current you inject is going to have to go somewhere to get to ground, and so it will go through the resistor or the capacitor. And from that, we can get the input current is G times V plus C dV dt. We're next going to move to having a battery in this circuit, in this equivalent circuit, in this conceptual model of the conceptual model. And where does the battery come from? The battery, the main battery, is thought of as a potassium battery. It comes from the Nernst equation, which I'll just present very briefly in a minute. And that's because you have a lot of potassium inside the cell and little potassium outside the cell. You also have a lot of sodium outside the cell, little sodium inside the cell. If you have trouble remembering that, just bite your arm, suck some blood, and it's salty. It's a lot of sodium chloride there. It's a lot of sodium in extracellular space. If you could get a tiny straw and suck out from the interior of the cell, it would not be salty. So there's a lot of potassium inside the cell, little outside. Potassium tries to come out. That's a diffusive force. It diffuses down its gradient. And eventually, it's going to stop. And what's going to stop it? The fact that it's positively charged and it's not being accompanied by negative charges, which stay inside the cell because they're mostly stuck to large proteins and can't move like that. So that electrical force eventually has to counteract the diffusive force, and that gives you a battery. And there are, I think, some batteries that are still built like this in a way. Right? I don't know. Anyway, here again, just a different picture showing the same thing. Potassium wants to come out, and as a result, it ends up setting up a very thin layer of positive charge on the outside of the cell, which gives you this electrical battery, which is expressed in millivolts, not volts, not kilovolts, just millivolts, little things, but big enough as far as the size of a cell are concerned. And that gives you an EK, a reversal potential for potassium of approximately negative 94 millivolts, which, if you only have conductance to potassium, would give you a resting membrane potential, or VM, of negative 94 millivolts. Here's the Nernst equation. I'm not going to go into it in any detail, just point out that this voltage difference is the gas, the Boltzmann parameter times temperature in Kelvin divided by the ion, the charge of the ion. In the case of potassium, it's 1. In the case of calcium, it's +2. In the case of chloride, it's -1. Times the Faraday constant, times the natural log of this ionic gradient. So the key thing to take out from the Nernst equation is that the electrical drive will be opposite and proportional to, well, not proportional because there's a log in there, sorry, to the difference in the ion concentration O outside, ion concentration I inside. So you can do a calculation with this. And actually, Salvador, I got this slide from you, and I tried to do a calculation on Neuron, and I failed. So I'll have to check back with you on that. Sorry, but we'll skip over this for now. What the potential is on the membrane depends on the dominant ion. A moment ago, we were saying the dominant ion is potassium, largely is, but at the peak of the action potential, transiently, the dominant ion is sodium. Again, biting and tasting, you see sodium is very high outside. And 150 millimoles is the approximation there, probably okay in most cases. Inside 10 millimoles, so from the nurse potential, you can calculate the reversal potential would be positive 60 millivolts. You remember the reversal potential for potassium was negative 90 millivolts. The resting membrane potential might be something like negative 70 or negative 80 millivolts, because it's a bit of a mix of both of these, as well as calcium. But during an action potential, potassium channels may close, sodium channels may open, will open, and that's going to give you this vast change in potential, which is referred to as a spike or action potential. It's active, it happens fast, a millisecond or so. In the mammal, more like a few milliseconds in the squid, but still very fast, fast enough to think with it. So we now get to these batteries and add those to the equivalent circuit. So we have this notion, as we were just saying, of extracellular and intracellular. We have the channels. Here we list EK as being negative 102. We said before it was negative 94, but again, this is not, it would depend on the cell. It depends on your, if you get hypernatremic or hyponatremic or hyperkalemic or hypokalemic. Actually, those are the words, famous, fancy Latin words for sodium and potassium. Hyperkalemia and hypokalemia tend to be particularly deadly. So potassium is really highly regulated. If you get to too high potassium, you're going to have seizures, cardiac arrhythmias, and death. But anyway, depending on your status of your particular cell, different cell types will be slightly different. You'll have a different EK. Again, there's a rule of what EK is, about 90, but it's also. Am I cutting off and on? This is, my little ear things keep telling me power off, which is discouraging. Presumably someone would tell me. Anyway, the overall membrane potential is this negative 60 and negative 75 is shown here due to these various different ion channels, each having different degree of permeability. One might ask, what maintains these batteries? Well, there is an ATP consuming, an energy consuming. Glucose makes ATP, which is the energy storage of the cell. Ionic pump to restore the battery. However, most of the time the battery is strong enough, you can go through quite a few action potentials before you need to worry about even having any restoration. So the ionic pump is not really needed for this whole mechanism, except for sustaining it in a more long-term basis. So we spoke a moment ago of conductance, G, the inverse of resistance. This is, again, a conceptual view of what goes on, which can be better described as permeability, P, which is the ion flow through these channels. The overall circuit up here includes the capacitor, as before, here in blue, the leak channel, and now, in addition to the conductance of this leak channel, we have thrown in the battery, and this battery is negative in. So cytoplasm's on the bottom, and you can tell a battery is negative from positive pole. Sorry, because the negative pole has the shorter line, and also in this case, the positive pole has a plus next to it. And then we have the potassium battery, which is going to be of variable conductance, and the line through the resistor symbol is that little sawtooth thing, the line through the resistor symbol with the arrow makes it variable, a variable conductor, or rheostat, the thing that you tune up and down on your wall when you want to dim the lights or increase the lights. That's a rheostat. Here, the rheostat is not controlled by your fingers, but is controlled usually by voltage. Similarly, the sodium channel, which is mostly turned off at rest, is variable and is controlled by voltage. And what it will do in brief is when you open that channel, it's going to put this battery into the circuit. The battery on the right here, so you can use this mouse. Actually, let me try this. This works better, sorry. The ENA will now be in the circuit and will tend to dominate, very briefly, the voltage across the membrane. So we can build up eventually to the parallel conductance equation from this diagram, again remembering Kirchhoff's law, which is that everything has to add up to 0 for conservation. It's not usable. Sorry, there's a sigma that wandered into this figure somehow. Ohm's law, definition of conductance again. V equals I over G, I equals G times V, but now with the, I use a different term here, reversal potential. Each of these has a reversal potential. So this is a reversal potential for E leak, might be minus 80, minus 70. For EK, it might be minus 90. For ENA, it might be plus 60. So reversal potential is kind of the generic name for each of these. Plugging it into Ohm's law, we have IL equals V minus ER over R, or G times V minus EL. Again, we have this inverse. This was the divided by R. This is times G. Again, we have the capacitor law. We change charge in coulombs into current in amperes. So we can say that the capacitance I equals C times dV dt. And then any current injected, and current here would be injected into the cytoplasm here, will be the summation of all the currents across these rheostats, across these resistors, across these battery resistors in series, and plus the conductance, plus the current through the capacitor. And so then we can just expand it out. G, the leak current is GL times V minus EL, sodium current, G sodium times V minus E sodium, potassium, G potassium, V minus E potassium, plus C dV dt. Back, front, there we go. So each of these equilibrium potentials is its Nernst potential, also called its reversal potential. Membrane potential is a weighted average, weighted by the conductances, and the resting membrane potential would be a steady state when the voltage does not change with time. For those who do not think a lot about calculus, I just wanted to give some notations because they tend to be used somewhat interchangeably. V dot means dV dt. V prime could mean dV anything, but here it means dV dt. Sometimes you might see partial derivative of voltage with respect to t, because we can also talk about a spatial derivative of voltage, dV dt, partial of v with respect to x. And when we do this numerically, we use the capital delta. That's a small Greek delta, that's a capital Greek delta. Delta V meaning a increment in voltage, which we could handle numerically as a small change in an increment of time. And in Neuron, we typically do 0.025 milliseconds, which is 25 microseconds. When you're at the steady state, all of these, which is just definitional, all these mean the same thing in this case, are equal to 0. There is no change in voltage with time. And so that's what's going to give us the resting membrane potential. And I will stop there and continue tomorrow and take questions if I can hear you, but I don't know if I can.
Thank you, Bill.
Okay, I can't hear you. Good. Only out of my left ear. Okay. So now if there were any questions on chat, I would see a chat. There's a chat box there. Chat. Nothing there. Okay. And I can see people here. So if somebody raises their hand, I will see that. And somebody like, if one of 9 people wave their hands wildly, I will see that. But this only see 9 people. I'm going to stop presenting, and then maybe I'll see more people. How to do that? You are presenting. That doesn't do anything. That doesn't do anything. Oh, there it is. Stop presenting. Now I still see 9 people. How can I see more people?
I think those are the ones that have the cameras on.
Okay.
All right.
Either
so there's no questions.
Extremely clear or extremely opaque. Good.
Okay, so we'll reconvene after lunch at 2 p.m. And I'll see you all in a bit. Thank you so much.